I'm your host, Dr. Ken Charlin, and today I'm very excited to have Dr. David Haase. Dr. Haase hails from the great state of Tennessee, where he has pioneered the Maxwell Clinic. Dr. Haase wears many hats. I've known him as one of the finest instructors I've ever had the chance to learn from through the Institute for Functional Medicine, and does excellent work as a consultant to other medical and biotech companies. But I'm going to save more of that bio because I know it's going to find its way into our discussion. I would say Dr. Haase is definitely a science and tech geek as great as they come, because you're going to hear about a lot of cool cutting edge things that he's up to. So we're going to call this interview Advanced Diagnostic and Therapeutic Strategies, and he is going to blow your mind. So, Dr. Haase, welcome to the Parkinson's Solution Summit 2.0.
Oh, thank you, Dr. Charlin. It's great to be with you. Thanks for doing such a great job pulling people together here.
Absolutely. And I brought you back for the second year of the Parkinson's Summit. We had a fantastic discussion about therapeutic plasma exchange, and there are going to be a lot of new folks here, a lot of folks who just want a refresher. So maybe we can start out with that, but I know there's some other areas that we want to get into, some really exciting emerging science and diagnostics that maybe we're still even a solid footing in terms of thinking about all the incredible uses that this technology may bring with it. And I think folks are going to be very, very interested in hearing about this. So let's talk about therapeutic plasma exchange and HOPE and what it is and how it can help people.
That sounds awesome. Sure. Well, plasma exchange is a very interesting technology. Now, all of us medical doctors have been exposed to it. It is the standard of care for very severe autoimmune disease. And I'm a qualified specialist in apheresis and very interested in this process by which you take blood out of the body, do something to it, and then put it back in the body. So what plasmapheresis or plasma exchange is putting an IV in one arm and removing the liquid part of the blood, which is the plasma from the cells, and then replacing that with a clean plasma replacement fluid, such as albumin, and then that all goes back in the body. And we have an individual sitting in a chair beside a very very technical machine. It takes a couple hours, but when you're done with that, we have removed the equivalent of all the plasma that you started with in the body. And this is really important because a big aha that happened in my life as a medical doctor happened because of this plasma exchange. I was going like that. And I need to rewind a little bit before I say just why this plasma exchange is so powerful for Alzheimer's disease and Parkinson's. But what really the interesting thing is that we know that this plasma exchange, in some of the research that we've done looking at longevity, actually helps the stem cells that live all over the body to actually behave younger. And since the stem cells are what create healing, if you change the environment of the cells, what we call the habitat, then the cells start behaving according to their habitat. And this was a big aha for me, that no matter what cells you have in your body for healing, wherever they are dependent upon their cellular habitat. And the habitat of the cell, you'd think of a bird, where their habitat's a tree in the outside, in the air. The habitat for a cell is what we call the extracellular fluid. This is this pond that hangs around all your cells, but all of your nutrients have to go through, all your oxygen has to go through, your carbon dioxide, your waste. And that's the interface between your cells and the outside world. And the part of this cellular habitat that lives inside your blood vessels, we call that plasma. So your plasma is this liquid extracellular fluid that happens to be all inside your blood vessels. And just like you are, your health is greatly dependent upon the health of your habitat, the air you breathe, the food you eat, the people that you're around, even the habitat of your thoughts and your ideas about your illness, right? That's your habitat. And that shapes your health. Same way with the cell. If the habitat is dirty or cluttered, if it has molecules that turn on inflammatory pathways, if it has molecules in there that turn on senescence, if it has all kinds of nasty molecular signals hanging out in this habitat, the cells are going to live in accord with how healthy that habitat is. So the remarkable thing about plasma exchange is that we can optimize the cellular habitat. We can remove the plasma that is in the body and then add in clean plasma replacement fluids. And then that extracellular fluid that's hanging out inside your blood vessels kind of migrates into around your cells. And the dirty stuff that's hanging out around your cells migrates back into the blood vessel. And then if we do another plasma exchange, we now clean the intravascular habitat, and then that can equilibrate with the cells that are in the body, your extracellular fluid. And the habitat gets cleaner and cleaner. And what's been amazing, I'm giving a talk for the Canadian Association of Apheresis Physicians in October, and I'm going to talk about how we've ignored this study called AMBAR. And AMBAR is a really remarkable study that looked at using this plasma exchange for the treatment of Alzheimer's disease. And it turns out that, and there's been a lot of subsequent observational data, but in individuals that have Alzheimer's disease, in a center in Spain that's been doing thousands of these, turns out that 70% of those people will have a halting of their progression. That's an amazing number. And that is supported by the AMBAR trial, which stated about the average person has about 61% less progression over 14 months. So plasma exchange has great utility, I believe, and we've been able to see it in the clinic as we do plasma exchange for people with cognitive decline and Alzheimer's disease. But the question is, can it help Parkinson's disease? And interestingly enough, my first patient ever in my clinic, a longtime patient of mine who was a cheerleader for me learning all of this stuff about apheresis, was a Parkinson's patient. And we brought him in. He did a plasma exchange. And the next day, he started to play the piano again. He went out and started a garden, which he hadn't done in 3 years. He took his wife on a date. And he went back, in that week, he went back and took over as chairman of the board of his company that he had been pulled away from for 3 years. It was a ridiculous transformation, which I think it had a lot more to do with hypercoagulability, and I think he actually had better brain perfusion. But I'm very fascinated with plasma exchange in the treatment of Parkinson's disease. And I'll tell you a little bit more about some diagnostics and why I think this has special applicability to people who have had COVID and where that goes from there. But that was kind of a whole lot of information really fast. But that's the redux on habitat optimizing plasma exchange for the approaching neurodegenerative disease.
I'm sure this happens to you a lot. It certainly does in my clinic, where preliminary data, things are done in the lab, mice experiments, maybe very early phase trials. Someone reads about that, and then the question is, well, should we be doing this in a more broad clinical scenario of treating Alzheimer's, treating Parkinson's, and so forth? And of course, the best standard acceptable answer is we need evidence, we need to do the big trials and all that. Why I'm saying this is, I know we've talked about this before, but I still find it fascinating, even though you're talking about humans. We want to quickly go back to that. I think those early experiments of sewing the mice together. If folks who are watching this haven't heard this, I mean, this is what Dr. Haase is talking about. I want to tell you, this science has really evolved. So he's at the cutting edge of it, but sometimes it's important to remind folks that there's a history here. There's a backstory, right? And that backstory has been around for a little while. It's just, how do we take that information and make it clinically useful? But I wonder if you just take a minute and talk
Sure.
about two mice being sewn together.
I think it's an incredibly interesting story. So actually, it starts before the mice. It actually starts with humans, interestingly enough, because what was noted by physicians like myself that are experts in apheresis, when they would do a plasma exchange on patients, sometimes for an autoimmune disease, sometimes other conditions would get better. Sometimes they got better from things that they weren't treating. And this has really been one of the big mysteries of apheresis. Why do some people have side benefits from this procedure? So that's a very interesting thing that happened long ago when plasma exchange first started for patients. But what was a very interesting set of experiments done by Irina Conboy at the University of Berkeley and then replicated at Stanford and MIT and Harvard. There's no question that this is real. You take cloned mice, an old mouse and a young mouse, and you actually sew them together by their side. Now, the poor little guys are now attached to each other as they run around the cage. And they share a microcirculation. So because they're clones of each other, they have the same immune system and everything like that. They can do that. There's no rejection going on. But what happens is a really interesting thing. That old mouse, within about 7 days, starts to turn young. The immune system reverts back to a younger expression. The fatty liver disease reverses. The osteoporosis that comes with age in mice reverses. The hair starts to be thicker. If a muscle is damaged, it heals faster, and the mice are able to become more muscular. The sense of smell of these mice returns. And in cognitive tests of memory of the old mouse, they improve their memory. So this was like, how on earth? And to understand this, it really does go back to understanding habitat. That old mouse, all their stem cells, all those cells that are scattered throughout the body that have the job of repair in a person, they, first of all, they go down drastically in number as we age. So the older we are, the fewer those stem cells are hanging out in our tissue ready to do the job of repair. But secondly, as we age, we also get metabolic clutter. We get molecular sewage building up. And that then creates a negative habitat for the stem cells, the old stem cells that are in the old mouse. But if you instead then create a young environment for those stem cells, they start behaving young and start doing the job of repair again. And so that was really remarkable. And then they went back and they said, well, is it all a lot of good things that are in the young mouse plasma that are doing this? So they decided just to use this clean replacement fluid that we use. And sure enough, much of the same effect happened if you do a plasma exchange on a mouse and just take out the old plasma and put in albumin replacement fluid. There are some other benefits that definitely do happen from the exposure to compounds that are in like a young mouse. So when we do plasma exchange, oftentimes there are opportunities for people to add in things that may be supportive of healing as well. So the whole science is very clear that this has to do with in many ways, the resuscitation of body-wide tissue-based stem cells so that they do the job of healing. And it's really just so, and I think what's so beautiful about this, and even if you or your family member don't have access to a clinician who can provide plasma exchange, one of the most beautiful things about this is it's proving everything we're talking about in functional medicine and systems medicine and lifestyle medicine, that the quality of your life habitat has a huge part to play on the quality of your body's ability to self-heal. And we're designed to heal. This has given me even more confidence and authority, really, in talking about all of those lifestyle factors that we do have under our influence so that we can change our genetic expression. So when we do plasma exchange, we're doing that just kind of all in a fell swoop, kind of like a snowblower clearing a driveway. But as we deal with our lifestyle stuff, that's actually slowing the snowstorm. And that's what we want to do.
But I'm so glad you're saying this because, you and I both utilize supplements. In fact, I don't know if you still are, but you're chief, I think, medical officer advisor for Zymogen, which, by the way, folks, very big supplement company, very fine supplement company. My point is that no issues with supplements, and they can be extremely useful as a tool in the toolbox. But it also tells us that if we're going to take this approach, that we're not going to fix the problem just by giving people a handful of supplements. They don't, and that's why my approach, when I meet with people, a very kind of Walls approach with Dr. Walls, I work very much together, but we have to start with the foundation. We have to, the first medical history is, tell me about your sleep. Tell me about, what you eat over the last couple days. Are you pooping and peeing? Are you drinking water? Tell me about your exercise. Do you exercise at all? If so, what do you do? I mean, we get all of those questions answered right from the front end. And I know that folks really love the testing and everything we do, and I'm sure that's the case in your clinic. And they're very anxious to get to that start line. What are my test results, right? But we're going to spend a month before we even talk about test results dialing in the foundation, or at least raising an awareness of how important this foundation is, even if we continue to evolve it and improve it and dial it in over several months. Because there's nothing that you can take that's going to overcome those kinds of hurdles if they exist. But what's very interesting that you're saying is that in some ways, well, maybe there is, but it's very holistic, and that is replacing the environment altogether, right?
Yeah.
You still got to do the work.
You still have to do the work, right? And no longer have that position with Zymogen. They're great companies, still use their stuff. And I think I always want to say, remember, supplements are supplemental. They are supplemental. They're not meant to lead the way. And that is, and in understanding why you're taking what you're taking, they can be incredibly powerful and helpful and useful. I'm really interested in a new molecule, one that's more commonly understood right now, called urolithin A. And it turns out that it's activating PARC and Parkin. So it's helping this mitochondrial or mitophagy at a substantial level. So I'm very interested in that in this whole discussion about Parkinson's and why, that compound naturally in the diet only comes up when people have a certain microbiome and they eat a certain food together. And not everybody, even some people eating the right food, don't make this molecule because they need the combination of the right food and the right organisms. And God, we're complex. It's beautiful. But we're never going to run out of things to learn. That's for certain.
I'm glad you brought this up. So let's segue into mitochondria and something that you lecture about, of course, and then how that relates to Parkinson's and this test that you can educate me as well as the viewers on the ME screen mitochondrial test.
Yeah, great. One of the big challenges, and I've been the lead faculty at the Institute for Functional Medicine Bioenergetics module for 10 years now. And I'm the guy who teaches the fundamentals of mitochondria, and I love that. Mitochondria, which are the powerhouse of the cell. I like that it has become a meme. I think that it's so important because every cell requires energy to do what that cell does. And the mitochondria is that powerhouse that enables every cell to do its job. And the mitochondria are like a big lava lamp, frankly. I like to have this joke that, how many mitochondria are in a cell? Well, the correct answer is one, right? Because it's just a big lava lamp that's kind of blobbing together. It's coming together in fusion and coming apart in fission, and it's always in movement. And this structure of the mitochondrial network is showing more and more importance. How the shape of the intracellular mitochondria have a lot to do with how they function. And mitochondria, you can imagine these are like, in many ways, they're kind of like the coal burning plants of the body. They're bringing in hydrocarbons, mixing them with oxygen to make energy, and you get carbon dioxide that comes out, but you also get soot and smoke that come out. And that soot and smoke, we call those oxidative molecules or oxidative radicals that cause oxidative stress. And, this is a kind of a dirty place in the mitochondria, when you think about it. And so there's lots of this smoke and soot damages the molecules inside the mitochondria on a regular basis. So the mitochondria has to continue to kind of clean itself out, so the engine is running clean. And one of the ways the engine runs clean is through this process of mitophagy or mitosis. So what we want to do is if you have a mitochondria, all the damaged parts are kind of herded over to one end of the mitochondria, and a pinching action is begun by some of the molecules, and it pinches off the damaged part of the mitochondria, and then that damaged part goes and gets digested. Well, it's interesting. So many of the conditions that are associated with Parkinson's are associated with this pinching off action. That mitophagy, which literally means eating the mitochondria, and so I've been very interested lately in this one molecule, urolithin A, which has a potential to upregulate PARC and some of these genes that induce mitophagy. So I think it's too early to make any conclusions about these compounds preventing disease, but I do think that they can be quite helpful. But the challenge is in measuring mitochondrial function, because the way you measure your mitochondria officially is you go to a lab, you have a muscle biopsy done at the lab, usually not using anesthetic, and they take a chunk of muscle and they run it across the hallway and they put it into a device called a seahorse, and the seahorse starts to analyze it from a metabolic standpoint. It will say how much ATP is generated, which is the energy molecule, how much oxygen is being consumed, how much lactate is building up, which is another kind of way of making energy. And then you can also measure these mitochondrial structural networks. You can measure how many reactive oxidative species are coming out. And that's a very difficult, painful test, and it's available in very few places, but that actually gives you a measurement of the function of the function of the mitochondria, excuse me. Well, in came the NASA. So many innovations happen when we send people into space. So they did a split twin experiment where they sent an identical twin into space and had an identical twin on the ground for a year. And they were tracking all kinds of biometrics. Well, they couldn't do repeat muscle biopsies up in space. So they said, well, we're going to have to find a surrogate way. So they developed this new blood spot card that would separate out the red cells from the plasma. It's a very specialized blood spot card. And they stored plasma and they stored cells. And they did this both on ground and in the space station. And then they came back and they looked at the difference between what happened with the astronaut that stayed on Earth and the astronaut that was in the International Space Station for a year. It turned out that shifts in mitochondrial function as measured by this platform were one of the biggest changes that were understood. So this change is basically using plasma to create what we call an imputed result, to create the habitat in a standardized cell line inside a seahorse machine. And then what that environment then shapes how those cells are going to behave. And we start measuring mitochondrial behavior as a result of the habitat. So I really think this is in many ways a great test because it's giving a clear understanding of the messaging environment and the intracellular habitat, or the cellular habitat that is shaping mitochondrial function. It doesn't necessarily measure the genetic abnormalities of the mitochondria, like electron transport chain uniquenesses. For that, I think our best, there's another test called mitoswab, which can be somewhat useful, but challenging to interpret as well. But this test called the ME screen, like mitochondrial efficiency screen, looks at multiple different parameters of mitochondrial function that can be tracked. And it's great. It's use a blood spot card. And interestingly enough, so it's highly accessible. And I really think that and it does take some challenge in interpretation of this. It is not a simple yes/no type of data that comes out, but it gives me great hope that we're getting to take a look at a true functional test. We're looking at, we're giving cells an environment and we're seeing how they respond from a mitochondrial standpoint. I think that's pretty exciting. So,
we're really at a pivotal time with Parkinson's disease. As we speak, there's been proposals about a new staging system, largely based on measuring alpha-synuclein in the spinal fluid using something called a seed assay or seed technology. It's even a couple papers that came out recently proposing that we completely ditch the term Parkinson's disease and refer to these disorders as I think neuronal alpha-synucleinopathies or something along those lines. But we're trying to navigate this, and folks, for watching, this is super important at Parkinson's because kind of like Dr. Haase was talking about the therapeutic plasma exchange and how sometimes we stumble on these observations and we pay attention. These things that happen sort of serendipitously are telling us something, and we latch onto it and investigate it, then it can grow into something much more significant. So in the story about Parkinson's, we have the infamous MPTP cases that came out of, I think, Los Angeles, California. I want to say it was the 70s. William Langston, the neurologist who probably was just a medical student or a resident at the time, but these drug addicts were showing up in the emergency room in a big clusters. It was all like 5 cases all at once, the same thing with acute Parkinson's disease, and they had inadvertently injected themselves with a mitochondrial poison. Now, they didn't, at the time, no one knew what that was. I mean, there was obviously an epidemiological investigation into that. And it turned out that some street heroin had been tainted with this toxin. But what happened as a result, first of all, and by the way, you can go onto YouTube and type MPTP, Parkinson's, Lancet, LANCET, because it was published in the Lancet, but actually watch the videos that were, because it was in print, but then there were additional videos that you could watch that went hand in hand with the articles. But these guys were given levodopa and they got remarkably better. So there was this aha moment that this isn't exactly Parkinson's, but it probably has something to do with Parkinson's or it's related. And that led to an understanding that MPTP was a mitochondrial toxin and how we could better understand Parkinson's disease by recreating this in the laboratory. And people like Malin DeLong, who recently passed away, but DeLong and his team from Hopkins and then Emory really mapped a lot of this out and figured it out. And their migration from Hopkins to Emory was because Emory had, at the time, the Yerkes Primate Research Center. So they were producing, MPTP monkeys and studying them and then, killing them and looking at their brains under the microscope, eventually leading to things like deep brain stimulation. But so important to understand that these mitochondria respond, guess what, to their environment. And that when you damage the mitochondria, that leads to a chain of events that we recognize as Parkinson's disease.
Mhm.
Makes me think, too, that with this emerging test, because there's we don't yet, in the traditional pharmacological sense, have disease modifying therapy for Parkinson's, like we now have 2 officially approved drugs for Alzheimer's. But we're close, we're getting closer, and I think that's why there's so much interest in creating these staging systems, because we have to know, is our therapeutic intervention actually doing something?
Mm-hmm, mm-hmm, mm-hmm.
We need the biomarkers, and basically what you're saying is this could be a biomarker.
Yeah. I think what's really interesting, and we're going to see a lot of the interactions with like the synuclein with the mitochondria themselves, because mitochondria are very much like a bacteria that lives inside our cells. It has much of those factors. I'm really fascinated with the infectious side of Parkinson's as well. I mean, like that MTPT data you were showing, the animal models of that, turns out doxycycline is somewhat protective against that. Doxycycline actually like inhibits the aggregation of alpha-synuclein in some of its more damaging areas. But anyway, I just think when I know this is the Parkinson's Summit, but we have to be really careful. Don't buy into the label Parkinson's too easily, because Parkinson's is the end state of a whole bunch of other things that came together in a very unique human. It's way more important to understand the person that has the disease than the disease the person has. And that's from Sir William Osler, one of our luminaries of modern medicine. And we can't forget that. So the diagnosis of Parkinson's or pre-Parkinson's should just be the beginning of your understanding. I love diagnosis, actually means dia is through and gnosis is to know. And so a diagnosis means to know through and through. It's not a naming and blaming. It should be an invitation into understanding. And as we understand, now we can start saying, hey, what are all the multifactorial factors that are perpetuating this process? Is there something we can do about it? There's another test that I'd like to tell you about that has a lot to do with this infectious side in a totally different way than most people have thought about it. And can I talk about that for a sec?
Yeah, please. That's great. Yeah.
So this has to do with a mystery that's been going on for a long time, because you hear people getting a whole genome analysis done. Do you know there's chunks of that whole genome that never get analyzed, never get sequenced? There's big chunks of it, and they just get passed over because they're so highly variable. Now we have these highly variable regions. One region is in this area where we make our B cells, and the other where we make our T cells. And this is how the body is able to do all this cool rearranging so that you're kind of ready for anything as you come out of the womb. Really, our immune systems are really miraculous, and they have a lot to do with these highly variable regions. But there are monstrous chunks of nuclear DNA that are also highly variable. And they've just kind of been like passed over. Oh, that's junk DNA. How many times have we done that? How many times have we, just because we don't understand something, like, well, it must not be important. So these turned out to be really important. And what Dr. Howard Ernovitz, a brilliant researcher, who I think should get the Nobel Prize for this work, has discovered endogenous ancestral nucleotide sequences. That's a big word for like ancient viral particles. Turns out that these highly variable regions have to do with the different chunks of viruses and other pieces of DNA that we have accumulated in our ancestry. So from one person to the next, different people groups will have certain types of these endogenous ancestral nucleotide sequences. And you go like, well, who cares? Well, this is why you should care, because we really think that this is in some ways like the operating system for the DNA. So we know that our environment causes a lot of changes in the expression of which genes get turned on. And we know there's lots of intermediate steps. Like if you eat sulforaphane, which is a compound in broccoli, that will turn on the production of the NRF2 gene cassette. And then you're going to get your genes that are associated with that gene cassette are going to start reproducing and make a bunch of antioxidant enzymes. And okay, look, environment had a lot to do with genetic expression. Well, but in many ways, we can't really make the connection between environment and why a certain disease comes up. It turns out that there are patterns that exist in these highly variable regions that give us the clue as to what end disease trajectory people are on. And so this one blood test, the early data, and we're the first center in the United States to be utilizing this. We've been partnering with the University of Iowa, University of Michigan, Vanderbilt's coming on board as well. This is super exciting technology. This one test can diagnose individuals with Alzheimer's, Parkinson's, MS, several different forms of cancer, and long COVID. And this is what's really fascinating because each one of these disease states are associated with a certain genetic expression pattern. Now, the research is early, and and we're participating in that research, and the funding is coming in. And anyway, it's incredibly exciting. But why I wanted to bring this up is that one of our patients who was actually coming in for plasma exchange, and we were doing a series of these tests before and after plasma exchange to see how quickly do these chunks of RNA reaccumulate in the body after plasma exchange. We want to know is this RNA always present, or is there parts of it that are there when we clean out the system with plasma exchange? How soon does it reaccumulate? What's the pattern like? Lots of really good basic science questions that we're answering here. But what we found is that we found some individuals that grouped with Parkinson's and also grouped with long COVID. And what this is really scary, honestly, because we have seen a big jump in early onset Alzheimer's disease. We've seen a big jump in cognitive decline since COVID. And more severe disease with COVID definitely perpetuates more neurodegeneration. One of the studies we're running here is the Evanthea trial, which is a randomized controlled trial looking at our precision medicine approach to reverse cognitive decline. And we're having to really pay a lot of attention to just when do people get COVID, because we think COVID is another big factor at driving forward neurodegeneration. So anyway, this test comes from a company called FBB Bio. That's like Fred Brown IO, FBB Bio. And it's very new. This is not something that, many folks are going to have heard about yet. But I think it's one of the most exciting things to help us understand this interface between environment and disease. I think with enough data accumulation, this is going to give us a lot of understanding about what in the environment is driving forth this particular genetic expression pattern. And so early is just diagnosing, putting a name on things. But later, especially with good data accumulation, I think this is going to have a big part to play in helping us understand what is most important to address for an individual, and then to clearly be able to track the effectiveness of that therapy, because these markers change with effective therapy. This is not just you have this disease or you don't have this disease. It is instead, are you on the trajectory of worsening of this disease process, or are you on the trajectory of improvement of this disease process? And I find that to be very hopeful.
these huge opportunities for me to learn as well as I do all the research and write these talks. And it really was very clear that if we sort of line up these big diseases, MS, ALS, Alzheimer's, Parkinson's, Huntington's disease, they all have something in common. And that is that, at least more downstream, the involvement of a pathological protein, a protein that's either been overproduced, misfolded, can't be degraded. We refer to this overall as proteinopathy. So the amyloid of Alzheimer's, the TDP-43 of ALS, Huntington protein of Huntington's disease. And even though most people don't think of MS as a proteinopathy, in fact, there's something called the bassoon protein that is part of the story of multiple sclerosis. So it makes me think there's a connection between what you're saying, because of course genes, DNA, RNA, protein, that's the progression. So it makes me think that what we're calling these proteinopathies, if we go more upstream, we're going to see these fingerprints that you're talking about.
I completely agree with you. And these proteins, like in Alzheimer's disease, the amyloid beta proteins, the p-tau 217, the alpha-synuclein, all of these in many ways have some antimicrobial activity to them. We think that a lot of these amyloid betaopathies or tauopathies, they come into being because the body own naturally produced antibiotics are becoming insufficient. One of those is LL37 or cathelicidin. And that actually has great homology. I mean, it acts the same as beta amyloid fragments. So I think infectious disease is a huge part of neurodegeneration. And I do think that we're going to continue learning more and more about that. The problem is the insult may be so much earlier than the later damage that we're now dealing with. Whether or not these conditions are persistent or not, we have a lot of work to do in that domain. Or if it's not just a fact, maybe it's just a small amount of infectious process there, but even the memory of that infectious process with an immune system that is now becoming old and untargeted, you now start getting friendly fire from the immune system. Our Department of Defense going out thinking it's trying to kill something it's supposed to kill, starts creating the injurious molecules that start damaging our own cells. But the great news is I think there's great progress being made on all of these fronts. Nobody ever wants a neurodegenerative disease, but there's never been a better time to have one.
Take-home solution, a solution, something here. We're talking about a lot of science, and certainly I know folks may want to seek you out for therapeutic plasma exchange because you are doing that in your clinic. Mm-hmm. Some of these other things are more exploratory, investigational, but it's still, the theme that's kind of gone through this whole discussion has been environment. And we know that plays a major role in Parkinson's. So if folks are going to do some self-help, some self-care as a result of this summit, they want to really put a magnifying glass. What can I change in my environment? Can I change my food? Can I make sure the air I'm breathing is clean? My water, my sweating, my pooping? And that may seem like an oversimplification, but these things are very powerful holistic tools that have profound effects, really beyond what a single drug can do, because a single drug targets one thing, right? Mm-hmm. Food targets many things all at once. Well, Dr. Haase, go ahead, please. Yeah.
Oh, no, just absolutely. Just couldn't agree more. But another interesting part about plasma exchange is I actually think it mimics the physiology of fasting and exercise. Because what happens with you're basically kind of running out of energy, you're running out of resources. When we do plasma exchange, the body all of a sudden is confronted with this need. It's like, hey, wait a second, a lot of the things that just used to be here aren't here. And I think it turns on its reparative structures in that way. So we have more to learn about we're always going to have more to learn. We know enough now to take action, but we're always going to be digging into the why behind it to continue to improve the process. So,
Dr. Haase, I'm looking at my notes for our visit today, and you've mentioned that you may have an event or at least a URL or an offer you want to share with folks.
if you'll just go to, you know what? I don't know. Maybe I do. Does it say where I was going to send people?
Yes, and it says we will have a PDF for this event. I'm assuming that maybe you want to share something, but certainly we can direct folks to your website and to read about HOPE, the Habitat Optimizing Plasma Exchange.
That's great. Sometimes I'm a way better doctor than I am, promoter in some ways. So, but come to maxwellclinic.com and please give our clinic a call, and we'll do everything possible to make certain that you get the information you need. So,
thank you. I think this has been a fantastic interview, and we're going to, have more and more excellent conversations, but we'll wrap this one up for today. Really appreciate you being part of the Parkinson's Solutions Summit.
thank you very much, Dr. Charlin. Appreciate you doing this. Thank you.